Hello everyone. So today we're going to discuss some of the applications of biotechnology in medicine. Recombinant DNA has made it possible for us to treat many diseases by replacing the damaged genes with new ones. So today we're going to take up some of the cases in which this recombinant DNA technology has played an integral role in providing great support and cure for certain diseases. First one is genetically engineered insulin. So, insulin is basically a hormone which is produced by the pancreatic cells. These pancreatic cells are known as islets of Langerhans. What does insulin do? Basically, insulin regulates the amount of glucose in the body. And people who do not have sufficient insulin in their bodies have a condition or a disease known as diabetes. Diabetic people are the ones which receive insulin as a treatment. This is important because without insulin, blood glucose will build up and the cells are basically starved of their energy source. So, it is really important to have that insulin in the blood. Insulin has two short polypeptide chains A and B. Here you can see in this diagram. So there are two chains A and B, and both of these chains are linked together by disulfide bonds. This is the structure of human insulin. So we have two chains A and B and they are linked together by disulfide bonds. Insulin is special in the sense that in mammals it is present in a prohormone form. So what does prohormone means? That this kind of form needs to be processed to be fully functional. So the prohormone form actually contains an extra stretch of C peptide. There is a three chains which are present, which is the A, B and C. So the C peptide has to be removed during maturation. Here you can see the proinsulin has the gray stretch, which is actually the C peptide, and after maturation the C peptide has been removed. So we have the insulin which has the two chains A and B which is bonded with the sulfide bonds. In 1983, Elli Lilly, which is an American company, found out a way to produce this insulin to help all the diabetic patients worldwide. Now let us see how they did it. The steps which are there in this kind of production require the extraction of that gene which is responsible for the production of the two chains. So the first step in insulin production is the isolation of human cells. These cells are then grown in cultures so that they reproduce and they proliferate. Then the gene segments which correspond to A and B chains are isolated. Because there are two chains, we try and isolate the genes which are responsible for both of these chains separately. Then the next obvious step would be that the genes which are responsible for A and B chains are engineered into separate plasmids. So both of these genes are then inserted into plasmids, and plasmids are separately transformed in E. coli. So we would have a colony of bacteria producing one type of chain and another colony producing another type of chain. Now after transcription and translation, we will get two separate chains of insulin A and B, and we will purify both of these chains. What is the next obvious step is that we have now two chains, but the human insulin will only be in the correct form when we join these two chains using disulfide bonds. So, following purification, A and B chains are assembled and disulfide bonds are created between them to create the actual mature human insulin, which will work inside the human. So, this is the insulin production. Now, let us move on to another very important way in which RDT has contributed to medicine. It is gene therapy. So, when scientists were thinking about how do you cure a disease which is genetic in nature, and is there a possibility of curing this disease completely? So, in gene therapy, they there lied an answer, a possible answer. What is gene therapy? We have to start with what are genes. So, genes are basic units of heredity. These are specific protein sequences. These are specific sequences which are which have the information to code for proteins. And when these gene sequences are altered, if there is a certain mistake in the gene, or the gene is out of normal, the gene has a sequence which is not normal to contain, the proteins which are formed because of these genes are not correct, or they do not function properly. This can lead to minor to severe problems in the person. So, gene therapy is basically when you utilize RDT to therapeutically deliver genes to patient cells to cure diseases. So, insertion of genes into individual cells and tissues to treat a disease is the premise of gene therapy. There can be many ways in which gene therapy can work. It can be inserting a new gene which is not there in the body. It can be inactivating a corrupt gene or a mutated gene, or it can be replacing it. So, we'll take an example here. We're going to talk about ADA deficiency gene therapy. So, ADA is adenosine diaminase. It is an enzyme which is involved in purine metabolism. So, ADA has its primary function as basically maintenance of the immune system. There can be many mutations with ADA. There can be severe production of ADA or deficient production of ADA. But we are going to talk about ADA deficiency here. So, ADA deficiency could be early onset or late onset. This disorder is caused due to the deletion of the gene which codes for adenosine deaminase. Now, there are a lot of possible treatments for this in the market, like the bone marrow transplant or the enzyme replacement therapy, which are conventional methods that are used. But they are not absolute cures for this. Gene therapy was seen as a possible treatment for this ADA deficiency. The procedure of this gene therapy for ADA deficiency would contain certain steps which we will discuss here using this diagram. So, initially, what is done is certain T lymphocytes are removed from the patient, right? And the T lymphocytes are then placed in a in vitro environment. Then in the lab, a retrovirus is altered and rendered in such a manner that it cannot infect a human. So it has no potential to possibly harm the human. Now this retrovirus is inserted with our therapeutic gene, which is lacking in the person who's suffering from this disease, right? So this retrovirus, now containing the gene, is inserted into the T lymphocytes. These T lymphocytes from the patient now become genetically altered. These altered cells are injected back into the patient, and these genetically altered cells can now produce the desired hormone. But here also, this kind of treatment will not last for a long time. This might last, however, if this kind of treatment is done very early at the embryonic stages. So, if I were to isolate the ADA gene and introduce it in the cells of an embryo, then this embryo will further grow into a child which could possibly be permanently cured of this disease. Now, as an exercise, you can stop the video here and try and solve these questions for your better understanding. Thank you so much, and I hope you understood these concepts clearly.